Hi, I'm Sandhya Prabhakaran and I'm an applied research scientist at the Integrated Mathematical Oncology Department at Moffitt Cancer Center with Dr. Alexander Anderson. Today at AACR 2023, we'll be presenting our work on understanding the progression of oligodendroglioma under the lens of normal oligodendrogenesis, and for this we use spatial transcriptomic imaging, single-cell RNA data and mathematical modeling. So oligodendroglioma is the second most found glioma after glioblastoma. And recent studies project that oligodendrocyte progenitor cells are the precursors for glioma, but this hasn't been explored in much depth. So what we do in this work is to test the hypothesis that the genetic mechanisms mediating oligodendroglioma are temporally and spatially coordinated. So oligodendrogenesis is the formation of oligodendrocytes, and these are highly myelinated cells, highly specialized cells that have a special way in which they differentiate, divide and progress. So for this, to understand the normal process of oligodendrogenesis, we use spatial transcriptomic imaging from VisGen. This is merfish data. So here we use the merfish image from a whole mouse brain. The cells are extracted, they are clustered. This is a UMAP showing the cell clusters, and this is the porting of the cell clusters on the spatial map of the brain. From here we extract the immature oligodendrocytes, the intermediate oligodendrocytes and the mature oligodendrocytes, and the genes that are used to annotate these clusters are actually validated from single-cell RNA-seq data, which is obtained from the mouse brain atlas from the Linnerson lab and CZI's Tabula Muris Consortium. We've identified the lineage tracing, the clusters using lineage tracing, and then use those genes to validate the clusters we have. Over here you see how we've actually used those genes that we get from both the imaging and single-cell data to actually form grids. Here showing the density of genes forming immature oligodendrocytes and mature oligodendrocytes. And this is then used into the mechanistic model that we've built to understand normal oligodendrogenesis. So we have a 3-compartment model that is to model immature, intermediate and mature oligodendrocytes. That is the cell diffusion, invasion and differentiation. And you can see this is how the model output looks like. This is the input from the single cell and the imaging data. And this is again the same case but for the intermediate cells. And we play that out over different time points, and the goal is to get the mature oligodendrocytes in a similar distribution that we see in the images. And you can see that these are very similar, so we're kind of happy with how we've established a mathematical model that explains normal oligodendrogenesis. With this as the base, we are now going to perturb the mathematical system to understand where the genetic aberrations form and occur, at what times they occur, and how this leads to oligodendroglioma. So these are some of our initial results, which say when we play with the self-renewal rates increasing and decreasing, we don't really get the same distribution of the cell types as from the imaging data. And this is again showing how we can perturb the system and use the mathematical models to simulate oligodendroglioma formation. So the future work for us would be to try and run this and validate this based on imaging data from the mouse brain that is actually malignant, and also to have this on 3D imaging, as well as better understand how glioma progresses over space and time. Thank you very much.